Good day to you, grade 8 learners. Welcome to today's Natural Sciences lesson brought to you by the Gauteng Department of Education. My name is Avezin Shwayo. In our lesson today, we are continuing with our area there on microorganisms. And today we want to focus on useful microorganisms. So what are our objectives for our lesson for today? Saying at the end of the lesson, you'll be able to identify the microorganisms that are useful in the ecosystem, right? So go through them. What are they used for, right? And then also you'll be able to explain how these microorganisms are used to make certain foods. I hope you are eager to know that as small as they are, these microorganisms, we as the humans use them to make our food in different forms. And then also you should be able to explain how the microorganisms are applied medicine. Yes, these microorganisms, they do have application medicine, and they've been responsible for saving lives for so many years in helping us to cure different kinds of diseases. Last but not least, to be able to discuss different ways of food preservation. We're saying know that our food get bad, get stale, so we need to know how we can actually save food, right? Saying all this is taken from our CAPS document, page 9. Right, so what is our pre-knowledge for our lesson today? Our pre-knowledge is our knowledge on microorganisms. Remember this we did last week in our previous lesson, grade 8 work on microorganisms. And we're saying at this stage, you are familiar with the fact that your microorganisms, they can be grouped into viruses. Remember, we said our viruses, they do not carry out all the 7 life processes. As such, sometimes can be considered non-living because they do not carry out most of these life processes. Then we spoke of the bacteria, which we said these are unicellular organisms. So we shall look at how these bacteria are useful. Okay, so how do human beings apply the bacteria different places? Then we've got the fungi. Okay, and remember we said our fungi they are multicellular. Typically, our molds. Remember we looked at that. Look at the molds that form on our bread. Those are fungi. Then last but not least, we also spoke protists, which is said like bacteria, unicellular organisms. So we're saying these are the groups of our microorganisms. We said in our previous lesson, our microorganisms, they cause diseases. And the microorganisms that caused named them pathogens. So now we're saying as much as they may cause disease or illnesses, uh, these microorganisms, they may find use, okay, as we shall see today lesson. Right, so to kickstart our lesson, we're going to begin with a small baseline assessment. Usual, I hope you are eager for these questions. Now for these questions, I'm going to give you 5 minutes. Right. Welcome back. Welcome back. Now let us complete our table together. Remember to join us on our WhatsApp platform 079 Okay, so now let us look at the table together then. So we have the different diseases there, types of microorganisms that cause them. So the first one, malaria. Remember, malaria is caused by a yeast parasite, and we're saying how is it contracted? Saying this is actually spread by female mosquitoes, call them the Anopheles mosquitoes, and specifically we give them the term vectors. They're the ones that they spread the malaria parasite. I believe we've had that experience with mosquitoes, a pleasant experience. So they're saying they're the ones that are responsible for transmission of our malaria parasite. Then we're saying the cells that are attacked are the blood cells. I hope you were able to get that correct and tick for yourself. And then the TB or tuberculosis, we're saying the type of microorganism, we're saying is a bacterium which causes tuberculosis. And we're saying this is through the inhalation of air that contains bacteria. We're saying cells that are attacked, those in the respiratory system, that lungs, some other organ cells. Right, so that you were able to get that one also correct. And then, so AIDS, which is caused by HIV virus, okay? And how is it contracted? We're saying this is contracted by through body fluids, okay? Body fluids. We're saying it is largely transmitted through contact affected blood, blood that contains HIV virus. And was saying the cells that are attacked there are the body defense cells, white blood cells, the immune system cells. Okay, so the virus actually destroys cells that protects the body from infection. Right, the last one there is our ringworm, saying this is caused by fungus, and we're saying that this is contracted through contact, mainly with objects that have been touched by affected person. For example, it can be clothes, it can be what? It can be blankets, it can be a comb, right? So we're saying, so it's mainly due to the contact with the objects that have touched the infected individual. Right, and what are the cells that are typically attacked? Saying these are the skin cells, also nails. Right, now that being said, I'm just going to give you time for you to make your corrections there. I'll give you 2 minutes for you to take down corrections. 2 minutes. Right, welcome back. Welcome back. So that you are able to read those directions. Right, let us begin our lesson for today. Let us start by defining few terms that we're going to meet as we progress with our lesson. First term, fermentation. We're saying fermentation is a process changing carbohydrates into acids, alcohol, and carbon dioxide. Can you take down that definition? Process of changing carbohydrates into acids, alcohol, and carbon dioxide. Right. Then the next term we want to find term decomposers. Right. We're saying decomposers are actually organisms that are usually bacteria or fungi that break down organic material. Right. So they break down organic material that includes dead plants, animals. Okay, so we shall see the typical examples of our decomposers there. Then our next term that we want to find, term antibiotic. I believe we are not meeting this term for the first time. Some of us we've taken antibiotics before. Okay, so an antibiotic, the medicine used to treat diseases that are caused by bacteria. Remember, our bacteria, saying they are also our microorganisms, which are harmful. But in today's lesson, we want to look at what useful kinds of uh, microorganisms. Right. So we use the antibiotics to treat bacterial disease. Right. Our next term, we talk of legumes. Can you take down that term? Legumes. We're saying these are a group of plants. Typical examples of these plants, they include bean plants, lentils, peas, and even peanuts. Right. So we're saying these plants, they have edible seeds or seeds that you can eat, okay, inside a pod. Think of the peanuts, they've got seeds inside the pod. Think of the peas, they've got seeds inside the what? The pod. And we're saying these, they fall under a group that we call legumes. Right, then there is preservation. What do we mean by preservation? We're saying this is the activity or process of keeping something valued, okay, alive, intact, or free from decay. So in other words, you don't want this thing to go bad, okay? So you want to keep it fresh. We call that preservation. We shall talk about it, the preservation of food in our lesson. And last but not least, penicillin. We were saying this is a common antibiotic. Remember we said our antibiotics, this is the medicine that we use to treat bacterial diseases. Right, so these are the terms that we are going to meet in our lesson for today. Are you ready? Okay, let us proceed. Right, remember we want to talk about useful microorganisms. Right, so we're going to start by looking at microorganisms that are useful in the ecosystem and how useful are they to us in the ecosystem. So our first group, we want to talk of the decomposers, and we say typically we have the bacteria and fungi. Right, so what is the role of these microorganisms in our ecosystem? We're saying they break down organic material. It can be dead animals, it can be dead plants, okay? So what will they do? They act on those dead plants and animals, okay? And in the process, they are going to release some nutrients into the environment, okay? So we're saying their role, they are decomposers, okay? That is our bacteria and our fungi. They act as decomposers, right? And we're saying they are going to break down the dead plants and the animal matter. Okay, so they break it down, And in the process, they are going to release some minerals into the what? Into the environment. Okay, And then we have another class of our microorganism. We are saying the protists. Remember our protists, okay? And we said our protists, they can be producers, okay? So they can manufacture their food from the process of photosynthesis, okay? And in the process, they will produce oxygen for our ecosystem. So we can see the important role now played by our useful microorganisms in our ecosystem. Right, I hope that makes sense. Let's take a closer look at our decomposers. Look at that image. Okay, we have that dead animal there. Okay, so our dead animal is going to be acted upon by our decomposers. Okay, and we're saying these decomposers, they have the function of breaking down that organic matter. Remember the energy flow in our ecosystem? When these animals, these consumers, they eat other animals, they get nutrients from those animals. And what happens when they die? Imagine if these decomposers were not there, it means the nutrients that are in these dead organic materials, they will remain inside them. But now, what our microorganisms will do, they will assist in cycling our nutrients. So when they break down, those nutrients that are reintroduced into the ecosystem, okay? And then they make our soil fertile. They break down the dead plants, they break down the dead animals, and that enriches the soil fertility. And remember, those nutrients, originally they came from our ecosystem, and they were now in the consumers. Now, since the consumers are dead, it is now the role of these decomposers to send back those nutrients into the ecosystem. So you see how important they are in keeping our soil fertile. Right, let us proceed. Right, I believe most of us have encountered that, or we have not really seen it at a close range when our parents, our sisters, our mothers, they are baking, okay, in the kitchen there. What happens? They normally use our yeast there, okay, and what happens is that the yeast will cause the dough to what? To rise, okay, when we are baking there. And what will be happening there, we're saying, the yeast actually creates carbon dioxide, okay? Now you know. It is the carbon dioxide that makes the dough to rise, okay? And in the process, okay, we have what we call a lactic acid bacteria, which is our microorganism that we use when we are making bread. And we're saying it gives our bread that sour flavor, okay? And we're saying, since it is an acid environment, the acid pH, I'm sure you know what we call by pH, right? The pH is low, and since the pH is low, it prevents the growth of pathogens. Remember we said our pathogens are our harmful microorganisms. So the use of these microorganisms, the useful microorganisms, it creates an environment that is not friendly to the harmful microorganisms and delays the process of our bread to grow stale. Have you noticed that our bread can go several days in good conditions without actually going bad? And we're saying it is as a result of these useful lactic acid bacteria which lowers the pH and hence makes our bread to actually stay long without decay. Right, so now we have mentioned about two uses now. Number one, we said our microorganisms, our useful microorganisms, they're important in our ecosystem. They act as decomposers. They recycle our nutrients, okay, to keep our soils fertile. And then we said now, when it comes to food, us as the human beings, we use this when we are baking our bread, okay? Remember the role of yeast. I hope you have mastered these 2 points for now. Let us proceed. Okay, our microorganisms, they also find role in the making of different kinds of food. Right, for example, bacteria can be used to brew wine and to brew beer. Okay, right, this is done through a process called fermentation, right? And then we're saying it is also used to make yogurt. I believe most of us love yogurt. We're saying that is done by the use of our bacteria. And also cheese. Okay, we're saying cheese is also manufactured through the use of our microorganisms, that is our bacteria. Okay, and so we're saying the process that is taking place there, we call it fermentation. And we're saying, remember, our fermentation, we're having the conversion of our carbohydrates, okay? And when we convert our carbohydrates, we can convert them into acids, alcohols, and carbon dioxide. So when we're making our beer there, we need this yeast, which is our microorganism. So I guess by now you are realizing that as much as we were talking about our microorganisms in our previous lesson, how small they are, that we cannot even see them by our eyes, by our naked eyes. But now you realize that even as small as they are very important, especially when it comes to the food that we eat and love. So take note of these products that are produced using our bacteria. Right. Also, we are saying our microorganisms, they're important in medicines. Okay. How important are they? We're saying these microorganisms are used to manufacture certain types of medicine. Right. For example, we talk of antibiotics. Right. We said these are the medicine that we use in the treatment of diseases that are caused by bacteria. Right. Let's look at a typical example of medicine. Right. Long time ago, there is a man called Sir Alexander Fleming. Okay. Sir Alexander Fleming, he discovered this, what we call our antibiotic. Okay. And this antibiotic is called penicillin. Please take down this information. Right. But I want you to look at how interesting that, how he discovered this antibiotic. Okay, he discovered it by accident. Okay, when he noticed that the bacterial culture that he made had been contaminated by a fungus. So it means he did not want this fungus, but he realized there is a fungus now in this culture that he was studying. So take note, this is the bacterial culture. Do you see this image there? And then inside you see that white stuff there. There is some fungus. Okay, this was not wanted in his research. Okay, but he realized there is a fungus. And then when he took a closer look on his culture, he realized that the bacteria which is surrounding, do you see that girl? The bacteria that was surrounding the fungus had been destroyed. Okay, quite interesting, right? So then he realized that this fungus was actually producing substances that killed the bacteria. Do you get it? So when he realized that this substance kills the bacteria, that's when he developed the idea of making this penicillin, which we are saying is a powerful antibiotic that destroys these bacterial diseases. So we're saying, what importance was that to us and to the rest of the world? We're saying the discovery of this penicillin, it changed the course of history. How did it change it? Remember, there were so many diseases long back that were not incurable, that were not curable, okay? And they were killing people. And through this accident by, say, Alexander Fleming, scientists were able now to manufacture the antibiotics, in particular the penicillin now, to cure these bacterial diseases. Quite an interesting story there, isn't it? So we're saying our microorganisms, they can also be used in the treatment, in the development of medicine for treatment of diseases. Right. How about microorganisms in plants? Remember, in our definitions, we spoke about legumes, okay, and we gave examples of typical leguminous plants like the beans and so on, isn't it? Right. We're saying in the roots of these plants, we actually find a bacteria there. Okay. Our bacteria is found in the root nodules of these leguminous plants. And we're saying, what do these bacteria do? Right. These bacteria, we call them nitrogen fixing bacteria. Right. So they help these plants to fix nitrogen into the soil. And we're saying the nitrogen is very, very important, especially when it comes to the manufacture of plant protein. Okay, so it's one of the nutrients that are needed by the plants. So these nitrogen fixing bacteria, which are called rhizobia, can you take down that name? Okay, so they actually live in the plant, and we're saying the plant is now the host. Okay, so what is happening there? We have a relationship that is happening there between the plants and our microorganism, that is our bacteria. And we're saying the kind of relationship that exists there, remember we call that mutualism. We're saying our microorganism benefits from living, from living in the plant being the host. And we're saying the host also benefits from the microorganism which fixes the nitrogen which the plant needs. And remember that relationship, we call it mutualism. So we can see that our microorganisms, as much as they are useful to us human beings, they are also useful to plants. Right? I hope that is making sense. Right. And then we're saying we also have microorganisms that we may find in the bodies of animals and even humans. A typical example, we have this bacteria called E. coli. Can you take note of that E. coli? And we're saying this is found in the stomachs of most mammals, okay, including humans, right? So there is an image for you to just have a slight picture of how it looks like. Our E. coli, okay, and we're saying this is found in the large intestines of many warm-blooded animals. Okay, and we're saying what is their main role? What does the E. coli help the mammals with? It helps the mammals by producing vitamin K2, and also it helps to prevent other harmful bacteria from growing in the gut. So you can see how useful our E. coli is. So now, at this stage, I believe you have been able to pick out a number of bacteria that are actually useful. Their role, how useful they are to the ecosystem, how useful they are to us, the humans, and how useful are they to the plants in general. Right. Now, at this stage, it's time we have a small activity on what we have covered so far. So I'm going to give you 5 minutes for you to tackle these questions. 5 minutes. Right, welcome back. Welcome back. I'm sure that we have successfully tackled all those questions. Remember your solutions, you can post them on 079 578 2908. So let us look at the questions together. You can mark for yourself the way you get the questions right. So, how are microorganisms used in nature and in industry? Right. Remember, we said in nature, we said they perform various functions such as the decomposition of dead organic material. And we said, importantly, that decomposition enables us to recycle the nutrients that were in the consumers or in the plants. When they die, they are now going to be taken back into the soil through the breakdown of those organic material by our microorganisms. Right. So that is their role in nature. And we said in industry they are used by people to manufacture different types of food. For example, we spoke of beer and wine, we spoke of yogurt, we spoke of cheese, which is the favorite of so many. And we're saying all those they are actually manufactured through the use of our microorganisms. Okay, and importantly, we said they are used to manufacture medicines or antibiotics. Okay, right. So I hope you were able to get those points. And then we said, who discovered penicillin? I believe by now you know the person. And how was it discovered? It's quite an interesting thing there. We said, Sir Alexander Fleming is the one who discovered penicillin, which we said is a very common antibiotic. Okay, and we said it was discovered by accident. It was discovered by accident. Remember, when he was studying his bacterial culture, he realized that it was now infected with the fungus. Okay, it was contaminated with the fungus. And then he realized that the bacteria that was surrounding the fungus had been destroyed. And from that, he knew that that fungus produced chemicals that actually destroyed the bacteria. And that's how he developed. That is how he developed the what? Penicillin. Right. Quite an interesting story there. And we say that this penicillin has helped the entire world over generations because we're saying this antibiotic has helped us to cure some diseases that were not curable before. Right. Now we want to proceed with our microorganisms. Oh, look at that side. I'm sure that we do not like that side. That shows us a rotting apple and some stale bread there. Okay, so I was saying, remember, our microorganisms are everywhere. We said these are a combination of harmful and some are useful. And we're saying they're everywhere. And now we're saying it is important that we avoid them contaminating our food. It leads us to the next aspect, which we call our preservation. Remember, we said preservation is about keeping what is valuable, that is our food. We want to keep it intact. We want to keep it fresh to avoid it from being stale. Remember, when food becomes stale, it has got that foul smell and even it tastes bad too. And we're saying if you eat that food, definitely you get ill. So the question is, how do we preserve our food? How do we keep it fresh? I'm sure that already in mind, we have got typical ideas or typical examples on how we preserve our food. Number one, someone is already hungry there. Right, we're saying the first one, we have a method called drying and dehydration. Okay, so we can dry our meaty food. There's so many kinds of plant material or even meat that we can actually dry. Okay, so we're putting this in the sun to prevent the spoilage by what? by our microorganisms. Can you please take note of these methods? Okay, so list them down. Dehydration. Okay, so we're saying this helps to preserve our food. It makes our food last longer before it is actually spoiled. Because we're saying our microorganisms, they thrive in wet or damp environments. So when you dry, like in the case of our meat, we dry it, we help it to last longer before it gets spoiled. Right, number two, yummy. Some of us, we are already recognizing that that is jam. Okay, so we're saying a combination of sugar and salt can actually be used to preserve food. Okay, why do they preserve food? It is because they do not allow the microorganisms to grow. Right, and remember, these typically are used in the preparation of jam, jelly, and even pickles. Right. So we're saying sugaring and salting is another method of preserving our food. Right. Number three. Okay, something that looks like my fridge there. Right. You can see we can store food that is perishable. All the food that you can see there is perishable. In other words, it can go bad. All right. So refrigeration can be used to preserve our food, and we're saying it is because of the low temperatures there which actually slow down the growth of microorganisms. And you agree with me that if you put food in the fridge, it does not mean that it will not go bad. It will eventually go bad. However, it takes longer to go bad because the low temperatures, they slow down the growth of the microorganisms. All right. Our next method that we want to talk about is called pasteurization. When you think of that, some of us, they think of milk, pasteurized milk. Right, and we're saying this is a method that we use to preserve our food. And we're saying, what does it involve? Pasteurization involves heating food. For example, it can be heated to 70 degrees Celsius. Okay, let's say it is milk, you heat it to 70 degrees Celsius for about 10 minutes, and then you quickly cool it, okay, to about 10 degrees Celsius. And what do you get? You get your pasteurized milk. And we're saying the process of heating, those high temperatures, they are going to kill our pathogens, okay? They kill our harmful microorganisms. In the event, we're saying you're also sterilizing the food. So you're getting rid of those harmful microorganisms. And we're saying our pasteurization is also a means of preserving our food because it prevents the contamination of our food by microorganisms. I hope you're taking note of these methods. So we are now on method number 4. Let us proceed to the next one. Okay, our next method is canning. Okay, canning our food. I'm sure we know of different kinds of canned food. And I'm sure that when you look at those canned foods, you've seen the expiry dates. The canned food, they can stay for years without actually expiring, isn't it? And we're saying what happens is that the food gets heated at very high temperatures, and then the food is stored at what? At airtight containers. All right. So the first step involves the decontamination. Okay, we're killing all the microorganisms before we put them into the cans, and we put them in airtight cans so that the microorganisms cannot survive. And we're saying this makes our food to stay longer without getting bad. Okay, so remember there we were saying our high temperatures, they kill the microorganisms. So that is number 5. Okay, let's proceed. Number 6. Right, we can use chemical preservatives. Okay, chemical preservatives. A common typical example of your chemical preservative is vinegar, right? Vinegar, this is a common acid. We can refer to it as acetic acid, or what is called ethanoic acid, right? Because it is an acid, we're saying it lowers the pH, okay? And the low pH, remember, we're saying it is not good for the what? For the microorganisms to survive or to reproduce, okay? So hence it can be used to preserve our food because it does not give the microorganisms a suitable environment for them to grow. Right. So this is our method number 6, and we're saying these chemicals. Remember, we normally put our food materials into jars there, and then what we do, we put our chemicals, and then our food is able to stay longer. It is actually preserved. Right, now that being said, this leads us to our last activity for the day. Right, for this activity, I'm going to give you 5 minutes. Welcome back. Welcome back. So let us look at the questions. Name 3 foods that are made using microorganisms. Remember, there is a number of foods which you can mention there. You can talk of yogurt, soy sauce, cheese, wine. That is not food. Right? Bread. Okay, that is also not food. But we're saying these are the kind of consumables that can be made in industry by people using the microorganisms. Right. And then the next part, the methods of food preservation, and describe how the food is preserved. So there is a whole number that you can mention. We mentioned drying. Think of biltong, which we do to our meat. You can even dry nuts, peanuts, and onions and other foodstuffs. And then we have sugaring and salting, which we said this does not allow the microorganisms to develop or to grow in our food. Refrigeration, we say the low temperatures, they hinder the growth of microorganisms. They reduce the rate at which they can grow. And then we spoke of pasteurization. We say think of milk. You boil the milk, you cool it. The boiling, remember we said that kills our microorganism. Then we spoke of sterilization in the line with canning there. Okay, remember sterilization, it involves the heating of our food products to high temperatures, and then you can put them in airtight containers. Okay, and hence they can last longer. And then we mentioned the chemical preservatives, the common one there being vinegar, which you say it is a low pH, which is not conducive for the growth of microorganisms and hence makes our food to last longer. Right, that brings us to the end of our lesson for today. So what did we learn in our lesson today? In general, we spoke about the role of microorganisms in our ecosystems, which we said they are decomposers playing an important role that is in the recycling of nutrients in our ecosystem. And then we said some microorganisms are used by people to make food. Remember the different kinds of foods that we mentioned, and then also to make medicines. Remember how penicillin was discovered. Right, and then lastly, we looked at the different methods to preserve our food to make it last longer. We spoke of freezing, drying, canning, and so on, and the different kinds of method. And remember, we are not learning these just for fun. Apply these methods and keep your food and make it last longer. Right, otherwise, that being said, I hope you found the lesson quite informative. Till we meet again, may God bless you.